Thank you for joining. My name is Alan Schroeder. I'm a clinical professor in pediatric hospital medicine and critical care, and I co-chair this seminar series with Dr. Rajni Matthew from Pediatric Infectious Disease. We launched this series last week, and I think a few things have happened since then relating to this pandemic, so lots to talk about today. Today we're going to talk about COVID vaccines, and we'll get to that shortly, and Dr. Matthew will introduce the speaker. Next week, we're going to talk about schools. And after that, we have a more didactic session by Dr. Arvin going over some fundamental principles relating to virology. If we can just get the next slide. This is how to get CME. If you like, you text code that number to the phone number listed there, and that will be a different code each week. So before we get to the main session today, we are going to start off each successive week with a presentation by a trainee. These will be evidence quick hits where we review an article of relevance that was published in the prior 1 to 2 weeks. And for this week, I'm thrilled to introduce Sunny Duong. He's a third-year pediatric cardiology fellow from here at Stanford. He's going to be leaving us next year to do an advanced imaging fellowship in New York City. But this week is going to be a little bit different because Dr. Duong will be presenting unpublished data. Most of the time we'll have peer-reviewed data published in medical journals, but we thought this topic obviously is receiving so much national attention currently that we thought it was worth presenting what little we know at this point, in part because we're already hearing about inquiries and demands from some of our patients and some of our families. So Sunny, why don't you take it away?
So I'll be talking about Regeneron's Regen COVID-2 antibody cocktail. I'm a PGY6 in pediatric cardiology. So this is not an FDA-approved use of this medication, and the data are not peer-reviewed. I sourced this data from the company's website and an investor webcast, actually. So as you all know, President Trump contracted COVID-19 about a week ago and was very quickly given this Regen COVID-2 antibody cocktail from Regeneron under a compassionate use request. And actually late last night, Regeneron asked the FDA for emergency approval for this drug that Trump on Twitter claimed cured him. So I thought it'd be very helpful to go through what exactly this monoclonal antibody cocktail was. So this is 2 monoclonal antibodies that were found to bind to the SARS-CoV-2 spike protein and prevent viral binding to the ACE2 receptor and allow the virus to be cleared without entering the cell. Non-human data from Regeneron showed that it can be used as prophylaxis and also help clear virus from the system better. Regeneron designed a seamless phase 1, 2, and 3 trial to test the safety, antiviral efficacy, and clinical benefit in symptomatic non-hospitalized patients. That's this trial that I'm going to be reporting, symptomatic non-hospitalized patients. Their endpoints were the viral load as determined by the nasopharyngeal swab, doctor visits, symptom relief, pharmacokinetics, and adverse events. And importantly, this is an interim analysis of the first 275 patients enrolled, but they're targeting more than 2,000 patients for enrollment. So adult non-hospitalized COVID-19 patients with symptom onset less than 7 days before randomization and a SARS-CoV-2 infection confirmed less than 3 days before randomization were randomized either to a low dose, 2.4 grams, a high dose, 8 grams, or placebo, and were monitored for 29 days after infusion. President Trump got the 8-gram dose, if you're wondering. So the mean age was around 44 years, with a large proportion of them being Hispanic, and the majority of them had greater than or equal to 1 risk factor for severe COVID-19. And didn't show the data, but the randomization will show that there is well-balanced baseline characteristics across treatment arms. They made a distinction between patients that came in who already had endogenous antibodies, those were that was seropositive, and those that did not have endogenous antibodies. And they found that patients that had endogenous antibodies had a lot lower viral titer in their nasopharyngeal swabs to start. And so they stratified the analysis by these characteristics. So in patients that were seronegative, the Regen COVID-2 antibody cocktail reduced viral load compared to placebo at day 7. It seemed to work better in higher dose compared to lower dose. And it seemed to work better in seronegative patients on the top bar compared to when you look at seronegative and seropositive patients combined. When you stratify them by their viral load, it seems to be that the REGN-CoV-2 cocktail is more effective at reducing viral load when you start out with more virus. And so you can see the difference here. Compared with placebo in the blue line, the treatment groups had a much greater reduction. They did try to quantify symptom relief, and in the seronegative population, there seemed to be an earlier median time to symptom relief in the treatment arms compared to the placebo arm, with the treatment arms having a median time to symptom resolution of around 6 to 8 days compared to 13 days in the placebo, but they did not provide any p-values for analysis here. I suspect that it's not statistically significant, but it's clearly different from the seropositive population. That when they stratified the groups by viral load instead of seropositivity, they found that there was faster symptom relief in the group with the highest viral load here, with a median time to resolution of symptoms of around 6 days compared to 13 days in placebo, and this was kind of borderline statistically significant in their analysis. They looked at doctor visits, and there were less doctor visits in the treatment arms, but there was a very low number of doctor visits in total, reflecting that a lot of these patients just in general got better. It was only 12 doctor visits out of 275, and these results were not statistically significant. So in conclusion, in a group of adult patients with symptomatic COVID infection in the outpatient setting, the REGN-CoV-2 antibody cocktail might be effective at decreasing viral load and alleviating symptoms versus placebo amongst patients who did not have evidence of serologic immune response or who had high viral loads at enrollment. And those two things are interrelated. It does not appear to work as well in patients who already have serologic antibody or who have low viral loads. There were less doctor visits compared to placebo, but that's underpowered. And there was no increase in adverse events compared to placebo. And I didn't show that data, but the adverse event rate was very low, equivalent to placebo. So I have a few unanswered questions looking through this unpublished data. What is the real meaning of seronegativity versus high viral loads? Which one is more important? And which one is the indication for treatment that we should be thinking about? And is there a relationship? There is a relationship between seropositivity and disease course. But are there patients that are late in their disease course, but for some reason aren't seropositive, that this might benefit them as well? How are symptoms measured? And is this really an important clinical endpoint for us? And are there other patient populations like exposure prophylaxis, hospitalized patients, critically ill patients where this would benefit? And indeed, Regeneron has other clinical trials where they're studying this exact question, but they haven't released any of that data. Thank you very much.
Sunny, that was great. Very concise review of some fairly limited evidence. And I want to put a plug in to a session that we actually have in November that will be led by Hayden Schwank from Pediatric Infectious Disease. It will be devoted entirely to therapeutics, and we may have a little bit more data on that then. I want to remind folks, though, that until then we have 0 data on children. And I also want to remind folks that to be mindful of using N's of 1 and potential misattributions of causality, and that a 70-year-old man who is obese, who certainly may be considered high risk, would still be considered to have a highly favorable odds of a complete recovery and a good outcome. And so we have to just be careful about perpetuating those misattributions of causality. That's just simple evidence-based medicine, which is what this series is all about. So with that, Rajni, I'm going to pass it over to you to introduce our speakers.
Thank you, Alan, and thank you, Sunny. Today, it is our pleasure to welcome 3 of our speakers who have each extensive work in vaccines as well as infectious disease. And I will introduce each of them before they start their talk. First up is Dr. Cornelia Dekker. She is a professor emerita with pediatric infectious diseases at Stanford. She's a consultant to SPEAK, which is Safety Platform for Emergency Vaccines. and is now focused on COVID-19 vaccine issues, such as developing consensus across the nations on issues like vaccine-enhanced disease. And she co-leads the Data and Safety Monitoring Board within SPEAK, advising on safety issues across different vaccine platforms. Thank you, Corey, and I will pass it along to you.
Disclosures, you've heard already from Rashni that I work for a mouthful of acronyms here. I am a consultant for the CEPI group via the Brighton Collaboration Safety Platform for Emergency Vaccines, or SPEAK project. I'm also a member of the Scientific Advisory Board for Medicago and consult with them on their influenza and COVID vaccine 19 candidates. So what I was hoping to cover today is give you a brief sense of what the COVID-19 landscape looks like, how COVID-19 vaccine development compares to vaccine development, a little bit about the statistical basis for the design of the United States phase 3 efficacy trials, and a comparison of the endpoints for those trials. So in terms of efficacy trial design considerations, SARS-CoV-2 and COVID-19 themselves are now only about 11 months old. Given that, vaccines are expected to become available within the next 6 months. We have limited, and that should probably read no time, for trial and error with these vaccine candidates. The phase 3 efficacy studies are complex in normal times. However, these are not normal times anymore. And no shortcuts are allowed to establish the vaccine effectiveness, safety, and trust, all of which are crucial for public acceptance of the vaccine that will eventually come out, or vaccine candidates, I should say. This is a busy slide. I just wanted to give you a sense of where we are. The slide itself is taken from an article in the New England Journal of Medicine, which came out in May 21st. And as you can see along the top, it goes and lists the technology, attributes of the vaccines, candidates in preclinical development, and candidates in human clinical trials. And just before this talk, I looked on the WHO website. There are now 42 different vaccine candidates in clinical trials. So this is a way outdated slide, but I'll use it still to give you a sense of what the landscape looks like. Representing DNA vaccines, Inovio Pharmaceuticals is in the clinic. We have inactivated vaccine candidates represented by Sinovac and Beijing. This is a platform that we know well because we have a lot of inactivated vaccines already. Live attenuated vaccines are being considered, and one is now already in the clinic. We have non-replicating vectors, protein subunits, replicating viral vectors, and lastly, the RNA vaccines, which are represented here by Moderna and the BioNTech and Pfizer vaccines, both of which are now deep into phase 3, and a host of others. Altogether, there are close to 200 vaccine candidates. So again, this is just a partial list, but to give you a sense that we really have quite a range of different vaccine candidates that may be coming through. This slide depicts the difference between, at the top here, normal vaccine development, and then on the bottom half of the slide, what's happening now with the COVID vaccines. And if we look first at the gray arrows here that are representing manufacturing and scale up, this happens rather slowly during the one foot in front of the other, phase 1, phase 2, phase 3 clinical development that is typical for vaccines. But you're not getting into large scale manufacturing until the licensure stage with a normal vaccine. In contrast, things are happening much faster, certainly with the clinical development, but the biggest difference really is shown here at the bottom with the commitment for manufacturing. So for the COVID vaccines, manufacturing development, scale up, the production of clinical trial material and validation processes happen very early. And by the time you're in the phase 3 efficacy trials, which for most vaccine companies and developers so far has happened within a 3 to 4 month period. There's already large-scale manufacturing that's ongoing. And this is really a huge financial risk that's being taken. It's worth it for the pandemic, but it's obviously not something that individual developers are going to do normally when they do this scale-up process, when they at least have some sense that safety has been cleared as a hurdle, even though they may not yet have the efficacy data which come at the end of phase 3. So that gives you a sense of what's different. There are, of course, a lot of other differences. Regulatory agencies are looking at these candidates and looking at data over time rather than waiting until the end where you submit a whole package. So all of that contributes to the fact that we have a pretty fast process ongoing, and it's specific for the pandemic because there are groups that have made this upfront risk in the manufacturing. So the COVID Prevention Network prototype trials, each of the phase 3 trials that's done per vaccine candidate has a population of about 30,000 adults that are at risk of SARS-CoV-2 infection and COVID-19 disease. There's not going to be pre-screening for this, so we won't know in the beginning how many people had already been infected, either symptomatically or not, although they shouldn't have had an obvious clinical disease. The randomization in these trials is either 2 to 1 or 1 to 1, and that's the developer's choice. And it's vaccine to placebo, obviously, and some are actually randomizing within risk strata. Follow-up is going to be for 2 years post-last vaccination, and the primary endpoint is virologically confirmed symptomatic disease. So here is a schematic of the follow-up and sampling schedule. As you can see, all the candidates so far are 2-dose vaccines that are in the clinic. So doses are given at day 1 and day 29. Then there are regularly scheduled clinic follow-up visits at which time blood is being taken to assess seroconversion. And then there are participant contacts for symptom-triggered NP swabs, anterior nasal swabs, or saliva cups that then are collected and tested centrally. And if there's a positive result, they enter into another aspect of the protocol that we'll talk about on this next slide. So for symptomatic cases that have a positive diagnosis of SARS-CoV-2, they will go into a 3-week, 21-day self-assessed symptoms with nasal swabs starting every other day for the first 10 days, and a blood draw that happens at the beginning of that process. On day 21, if the SARS-CoV-2 test is still positive, then they continue follow-up with these nasal swabs and another blood sample at day 28 until resolution of symptoms. If they're negative at day 21, then they come back at day 28 for blood draw and nasal swab. So looking at the endpoints, here is the common primary endpoint for all of the phase 3 efficacy trials, and it's COVID symptomatic infection. Virologically confirmed, you have to have symptoms in order to trigger this. There are, however, some key secondary endpoints: positive SARS-CoV-2 PCR or seroconversion defining all SARS-CoV-2 infections. And over here on the bottom, as a branch of the COVID symptomatic infections, the trials will be looking for severe COVID disease as well to determine efficacy against that. In terms of when the primary analysis will be happening, this is event driven, and for the trials that are randomized 2 to 1, they'll stop for the primary analysis after 150 events have accrued. The 1-to-1 ratio will have 170 events. The trials are sized such that with conservative assumptions, this primary analysis is likely to take place within about 7 months of the start of the trial. Continued blinded follow-up, however, after this 7-month time point, roughly, is going to be necessary in order to evaluate the duration of vaccine efficacy, which is a secondary objective, and to adequately power the secondary endpoint of vaccine efficacy against severe COVID disease. Because that's a subset of all of these events, these are all symptomatic, you need to have longer follow-up in order to accrue more cases to get this vaccine efficacy endpoint of severe COVID disease. This is my last slide. This is what happens depending on what the outcome of the interim and primary analysis shows. If the efficacy criteria are met, shown here on the left, they will stop randomizing cases. The blinded follow-up will continue. The result is reported publicly, and a select group of investigators are provided the unblinded data so that they can produce reports. Over on the right is the worst case, no efficacy, or in fact, if there's an increased risk of disease that gets shown. Again, randomization stops. The oversight group determines whether and how long to continue the blinded follow-up at this point. And if demonstrated vaccine harm or a major concern is determined, then the participants may be unblinded and notified so that they can obtain adequate follow-up. And lastly, that result also was reported publicly. In the middle is perhaps a more likely scenario here. No criteria are met. Well, that's easy. Randomization and follow-up will continue as planned for these trials. Thank you. With that, Roshni, I'm going to set it back to you.
Thank you, Corey, very much. There was just one question that hopefully we can just take it now. It was about the definition of symptomatic disease. Does that vary between different trials, different vaccine trials? What is the definition of symptomatic?
Yeah, there are some differences in that definition, but they're at the margins, I think, is what people would say. Obviously, there is the respiratory distress, shortness of breath, fever, cough. Any of those occurring will be fairly common, I think, amongst all of the trials that are ongoing. But there are some slight variations in terms of how the developers have actually defined that definition. And this is what they discuss with the regulators and what gets approved in their final protocols.
Thank you, Corey. And we'll take questions at the end, but we will move on now to our next speaker. I am very happy to welcome Dr. Nicola Klein. She is a board-certified pediatrician. She's the director of Kaiser Permanente Vaccine Study Center since 2006. She's a professor in the Department of Health System Sciences at Kaiser Permanente Bernard J. Tyson School of Medicine. She has worked extensively in vaccine safety and effectiveness and will be leading the Vaccine Safety Data Link Rapid Cycle Analysis, which will be monitoring COVID-19 vaccine safety. She is the principal investigator on numerous vaccine clinical trials and is currently the PI for Pfizer's phase 3 clinical trial evaluating the safety and efficacy of an mRNA COVID-19 vaccine. Thank you, Nikki. I will hand it over to you now.
Thank you very much, Roshni. Advance the slides. There we go. Okay, so here are my disclosures. I do receive funding from Pfizer for the mRNA vaccine trial, as Roshni mentioned, as well as funding from almost all major vaccine manufacturers for unrelated studies as well. So for today, the goals of the presentation are really just to give a very bird's eye view and 1,000-foot thoughts about the U.S. vaccine safety monitoring system, and then to briefly describe the plans for monitoring COVID-19 vaccine safety. So just a brief word that when you think about vaccine safety, we do actually have to think about it a little bit differently than we do about drug safety, in that there is a higher standard of safety, typically because vaccinees are generally healthy and maybe very young, is particular to this audience, as you are well aware. Vaccinations may be universally recommended or mandated. And just for all these reasons, there's just a much lower risk tolerance for example, a vaccine adverse event, something along the lines of less than 1 in 100,000 for an adverse event, versus the risk for a drug adverse event, which may just vary from everyone getting it depending on the indication for the drug, up to 1 in 1,000, for example. Now, we just heard about some of the pre-licensure studies, which are pivotal and key for both efficacy and safety of any new vaccine. And we just heard some of the some of the protocols for the randomized blinded placebo-controlled design. And these are wonderful advantages. There's close detailed follow-up. And of course, they're blinded and they can be controlled. However, there are disadvantages in that the rare or delayed reactions are often can be difficult to detect. And then typically the vaccine trials do not necessarily include subpopulations, so we won't know what happens with those subpopulations. So when you start to think about vaccine safety, I think that probably everyone on this call is quite aware that for the need for ongoing vaccine safety programs. So this is this classic graph from Bob Chen, published in the early 1990s. And typically, when I show this slide, I usually like to, I hope you can see my pointer, we usually are thinking about what happens here with vaccine safety, because we're worried about increasing vaccine coverage, and there may be a loss of confidence, and then there's an outbreak of a disease because of decreased vaccine coverage. And so this is usually the place at which we start to think about vaccine safety and as it affects public health. However, right now, I think we are in an, as you know, not to be too cliche, but the unprecedented time of thinking about COVID-19 vaccines, vaccines and vaccine safety here at this point, which we don't even have a vaccine yet available, but it's to be anticipated that there probably will be one soon. And so we're here trying to get increasing vaccine coverage in the face of this pandemic. However, at the same time, what we're having here is already a questioning and a possible loss of confidence. And so we might be having these two things happening at the same time. And so this is just really highlights and reflects the need for even more enhanced safety monitoring for COVID vaccine safety, so that this actually doesn't come to pass. So in terms of generally, what is generally the post-licensure vaccine safety monitoring? So I've highlighted it. The ones in asterisks are more enhanced vaccine safety for COVID-19, but most of these are actually things that are already in place that we've been doing for a long time. The first one is that the CDC has a very pivotal role in vaccine safety monitoring out of what's known as the Immunization Safety Office, and that includes the Vaccine Adverse Event Reporting System, also known as VAERS. There's something new called VacSafe, which is for COVID. I'll talk about that in a minute. Now there's the Clinical Immunization Safety Assessment Network, and then the Vaccine Safety Data Link Project, and I'll talk about those briefly in a few minutes. The FDA also does have a role for post-licensure vaccine safety monitoring, and currently, right now, it incorporates the Sentinel initiative, which is an insurer and payer database, and that focuses mainly on drug safety, but they also do some vaccine safety as well. And this is typically 100 million covered lives. That's what the number for Sentinel usually includes. And then in addition, there's what's called BEST, or the Biologics Effectiveness and Safety Program. And this is CMS data monitoring, which includes 650,000 long-term care facility residents. Now, finally, the vaccine manufacturers also have a role in vaccine safety monitoring, and typically these are in the form of an FDA-required phase 4 vaccine safety studies. These can typically be on the order of 10,000 up to 50,000 people or more. And these are vaccines that are monitored as part of safety monitored as part of routine vaccine use. And just as a note, we've done quite a few of these studies within Kaiser, which we look at the data for people who receive vaccines as part of routine care. So spending a minute on the vaccine adverse event reporting system. So this is a passive system. It's co-managed by the FDA and the CDC, and it's been around since 1990. This is web-based reporting for suspected adverse events, and this reporting can come from physicians, patients, etc., sometimes even lawyers. But there's also public access to the data. Anyone can report. Anyone, you don't have to justify why to do it, and it's for anyone who thinks there's been an adverse event for any reason. It's national in scope. It's very timely, in particular for the COVID-19 enhanced reporting. There'll be the report processing times will be daily data sets, and the reports will be evaluated within 1 day for death reports, within 3 days for serious reports, and 5 days for non-serious reports. It's considered a generation of hypothesis for generating signals. And it's going to be part of an enhanced VAERS reporting using the National Healthcare Safety Network, or NHSN sites, which will be facilitating reports for healthcare workers or long-term care facilities. And again, this is part of the enhanced vaccine safety monitoring in addition to normal VAERS reports that happen. So this is one of this is what that vSafe I mentioned briefly, is that this is an enhanced COVID vaccine response. This is vSafe is a smartphone based text to web survey, and email to survey. They will be actively surveilling early vaccine recipients, mainly essential workers and healthcare workers. This has been estimated to be ready to monitor for up to 20 million vaccine recipients in the early months, and there'll be text messages or emails from the CDC for vaccine recipients, and then anyone who reports any possibly clinically significant event will get reported to VAERS, and then they will follow up also with the healthcare workers themselves. So a brief word about the CISA network. And this was established, and this is again managed by the Immunization Safety Office. So this is established in 2001 as a national network of medical research centers with expertise in immunization safety. The goals are to improve understanding of adverse events following immunization at an individual patient level, to assist the CDC in evaluating emerging vaccine safety issues. And most relevant for current times is that it will serve as a vaccine safety resource for you healthcare providers with complex vaccine safety questions about specific patients to assist with immunization decision making. And this has been an activity that CISA has been performing for a long time, but right now, as part of the enhanced CISA COVID-19 vaccine clinical consult service, there will be 24/7 coverage availability for clinical consults for vaccine adverse events. Now, a word about the Vaccine Safety Data Link, also through the Immunization Safety Office. So this is a collaboration between the CDC and 8 integrated healthcare delivery systems, of which you'll note that Kaiser does comprise the majority of these sites, but not exclusively. It started in 1991. It includes about 10 million people, which is usually about just over 3% of the American population. And what the Vaccine Safety Data Link, or VSD, is an active surveillance of newly licensed vaccines. It also evaluates the vaccine safety of new recommendations for existing vaccines, for vaccine at high-risk populations, particularly pregnant women and other groups as well. It also develops new methodologies for vaccine safety assessments. And finally, it also it's a system for testing hypotheses noted by signals from VAERS, from clinical trials, and from other platforms that signals might come up. And it just conducts a lot of vaccine safety research in general. So one quick word about a particular type of vaccine VSD studies, what's known as a rapid cycle analysis. And this is an ongoing surveillance of targeted outcomes. And this has been some of the VSD's been conducting for all newly licensed vaccines since about 2005, 2006. And it gets weekly updates of all the data from all the sites for the VSD. And that data is evaluated on a weekly basis for those targeted outcomes of interest. Now, I've just included an example of the measles, mumps, rubella, varicella, or MMRV vaccine was the first example of an RCA detected safety signal in which we identified increased risk of febrile seizures 7 to 10 days after MMRV as compared to MMR plus V. And you can see this in the graph below, in which you see that the blue line indicates seizures after MMRV, and the pink line is the seizures 7 to 10 days after MMR plus separate varicella vaccine. And so this has, since this time, and there's been many vaccines that have been monitored using these real-time updates, and this has become really one of the key and very valuable tools that the VSD has in terms of monitoring in real time new vaccines as they are started and being rolled out. And for COVID vaccines, the planning's already underway to start to plan so when the first vaccines are given within a population, the rapid cycle analysis for the COVID-19 vaccines will be ready to go on day one. And we can start to monitor and look at data weekly for outcomes of interest. So in this last slide, I'm just giving you a list here. So this is a long list, it's very busy, but I just want to give you an idea. These are the particular studies that are currently either confirmed or tentatively planned that the CDC is sponsoring for COVID vaccine safety. There's going to be the rapid cycle analysis I just mentioned. There's vaccine safety in pregnant women and their infants. There's going to be vaccine enhanced COVID-19 disease monitoring. There's some evaluations of changes in healthcare utilization due to the pandemic and how that might impact vaccine adverse events encounters. We're going to be monitoring both influenza and COVID-19 vaccine coverage in pregnant women. And then finally, looking at COVID-19 disease in pregnancy and fetal death. And then I mentioned CISA will be doing an enhanced 24/7 clinical consult service for vaccine adverse events. And then there's a list of potential VSD and other CISA studies, which I won't go through in great detail, but I just want to let you know those may or may not still come to pass, but that's what is potentially on the docket right now. And this is, I think, with that, I think I will stop and turn it over.
Thank you very much, Nikki. We have a lot of questions coming in, and we are going to hopefully get to some of them, at least many of them, after Grace's talk. I would now like to welcome Dr. Grace Lee. She is a professor of pediatrics, infectious diseases at Stanford. She's also an associate chief medical officer at Lucille Packard Jones Hospital at Stanford. She's a board member for the Society for Healthcare Epidemiology of America, or SHEA. She's also the board member of Pediatric Infectious Disease Society and the National Academy of Medicine Board on Population Health and Public Practice. Dr. Lee is the vice chair of CDC's Advisory Committee for Immunization Practices, or ACIP. She's a member of the COVID Vaccine Working Group and the chair of COVID Vaccine Safety Technical Work Group. Welcome, Grace.
Thanks very much. So I was tasked with perhaps just walking this group through the process, and happy to talk about what's the same or different. But I've actually been surprised that a lot of people, and perhaps maybe because we live in a pediatric world, many of us know what the Advisory Committee on Immunization Practices is. But I share these slides in part because there are many individuals who have never heard of ACIP until COVID vaccines hit. So it's been an interesting time. It was just clearly articulated by Dr. Dekker and Dr. Klein, there's a phase of vaccine development and testing. We're still clearly in the phase 1, 2, and a few vaccines are in the phase 3 clinical trial stage. And typically what would happen is that the submission of that data would go to FDA for a BLA, or Biologics Licensing Application. As many of you know, much of what's been discussed lately with regard to COVID vaccines is whether or not it would go under emergency use authorization or would go for full FDA licensure. The typical process, again, after FDA licensure, there is a independent, and I mention this because there's actually 2 independent committees that are federal advisory committees, and VRBPAC actually advises the FDA and ACIP advises the CDC. VRBPAC really focuses on the regulatory use of vaccines and thinks about the benefit risk assessment of any biologics, and in this particular case, vaccines. And ACIP generally considers the use of those vaccines in the US civilian population. So after FDA licensure, it would come to ACIP for consideration, and ACIP makes recommendations about use, which typically then go to the CDC director and get published in MMWR. And then of course, there's all of these other logistical considerations which are actually huge this time around with COVID-19 vaccines, but financing and implementation, and in particular implementation, will be the challenge. Just to reiterate that ACIP has been in place for a very long time. It was established in 1964 to provide advice and guidance to the director of the CDC on the use in civilian populations based on these particular characteristics. So we consider disease epidemiology, vaccine safety, vaccine efficacy and effectiveness, the quality of the evidence reviewed, economic analyses, and implementation issues. And I think what's a little bit different about ACIP compared to any other federal advisory committee is that we actually do consider cost effectiveness as one component. That clearly will not be the driving component at all for COVID vaccines, but it is unusual in that we actually consider that part of our mandate. I bring these up just because, again, most people don't realize where the recommendations for child, adolescent, and adult immunization recs come from. So these are the recommendations we use every day as pediatricians, and our adult colleagues use as well. Just to put it in context of this is typically our process. We have 15 ACIP members with various expertise and perspectives that are listed here. And you can see that's the group at the inner table. And at the outer table, I'll highlight that we actually have 8 ex officio members from federal agencies and 33 liaison organizations that are all involved in vaccines in one way, shape, or form. And I'll, for example, Bonnie Maldonado, our division chief for infectious diseases, is there for the AAP and plays a very important role. In February of 2018, so I will say since 2012, actually, ACIP adopted what's called the GRADE framework for considering the benefits and risks of any particular vaccine. But in addition, we ended up adopting what's called an evidence to recommendations framework because there was a recognition that decision-making is a complex process, and depending on the 15 members that are sitting at the table, you could be given the same evidence and come to different decisions about use of a vaccine in the population. The ETR framework was adopted in order to enhance transparency in decision-making, consistency of the implementation of the approach we use, and specifically to communicate deliberations and judgments made during the formulation of recommendations. In particular, it's been helpful to have this tool as a way to express dissenting views or minority opinions. And these are the components of the evidence to recommendations framework that we use in ACIP, or we have been using since 2018. And we are going to lean on this absolutely for COVID-19 vaccines. So typically, we ask about the problem. Is a problem of public health importance? We think about the benefits and the risks. So, is the intervention, what is the magnitude of the benefits, the magnitude of the risks, and the balance of those, in addition to what is the certainty of our knowledge about those benefits and risks. We think about the values of those who are being vaccinated. And as you know, it's going to be a really important aspect of being able to deliver vaccines with COVID-19. We take a look at acceptability. So is it acceptable to key stakeholders? And actually for this, it's actually related to ethically, programmatically, and financially. And you can see in particular with regard to ethical frameworks for vaccination and early vaccine allocation, this has been a really robust and terrific source of having conversations about some of the inequities that we've seen in our health system. We also consider resource use, usually cost effectiveness analysis, and then feasibility. Is it feasible to implement, and are there any barriers to implementation? And again, a huge consideration. What's implicit in this framework is equity, but I will say that what COVID-19 has done for us, at least, is obviously this has got to be explicitly incorporated, equity as a principle in any vaccine recommendations. And in fact, I think we're rethinking about how to actually build in equity explicitly into our evidence to recommendations criteria. But for COVID-19 vaccines, it is clearly an overarching, and in almost every single one of these domains, something that's incredibly important. So these are the work group members for the COVID-19 vaccines. So you'll see here that we have a large group of individuals. We have been meeting, essentially the work group started in April. We initially met bi-weekly and we're now up to weekly. And in addition to this, Roshni mentioned, although I didn't put the slide up, that there is a vaccine safety technical subgroup that I chair that specifically is looking at all aspects of vaccine safety, pre-authorization or pre-licensure, as well as once a vaccine does come to market, then we will take a look at all of the safety data in that post-market phase. So I mentioned that the work group is meeting weekly. We've already reviewed phase 1-2 data from manufacturers as data become available, and we are looking forward to reviewing the phase 3 data when it becomes available. Once that data do become available, which, there's been a lot of discussion about when that would be, we do anticipate conducting an independent review of the safety and efficacy data. We'll use the evidence to recommendations framework and the grade process in all of this. And then the full ACIP will hear all of the options based on the work group deliberations. We anticipate having an emergency meeting with a public comment session whenever the FDA decision is announced. And we'll vote on recommendations for vaccine use then. I think one of the challenges we've run into is that implementation of COVID vaccines is going to be complex. It's not like anything we've ever seen before, in that there are so many vaccine candidates with different storage and handling considerations, with different numbers of doses that are using different platforms. So the sheer implementation challenges are of course weighing in, are thinking about the recommendations in addition to the safety and effectiveness. And then after an ACIP vote, we'll submit our recommendations to the CDC director. I think what's unusual in this case, let me just see if there's another slide here. I think what's unusual in this case, let me do one more thing. So in terms of the path to COVID-19 vaccine recommendations, this was just on Tuesday. This was literally what happened like in 12 hours on Tuesday. Actually, Monday night, they published that the White House blocks the new coronavirus vaccine guidelines. So for those of you who have been following the news closely, in June, July, actually, FDA came out with specific guidance around the characteristics of the COVID-19 vaccines that they wanted to see. So for example, efficacy of 50% with a lower bound of 30%. And I actually think that guidance was excellent and extremely helpful for aligning some of the trials that are coming through now. And in addition, we had been waiting for most of August and September for the FDA guidance to come out that apparently did get pushed out on September 21st with regard to other considerations, in particular safety and what FDA would be expecting and how much follow-up would be needed. So the White House actually blocked it for 2 weeks. Then FDA released it via the VRPAC meeting. So that if you wanted to take a look at what their guidance is, take a look at the anticipated October 22nd meeting. It's a public and open meeting, and the Federal Advisory Committee for FDA will be deliberating or bringing that document into view. And then later that night, the White House reversed their decision, and they decided to go ahead and accept the FDA guidance. So it's been a little bit dizzying to see what's going on. I kind of just have to wait for the news cycles to kind of pass through to figure out where we need to head. But I think in general, our goal has been to focus on staying the course, taking a look at all the evidence to date and and thinking about our recommendations. And I'll point out that the one unusual thing about this particular framework that I have not experienced before in my 4 years on ACIP has been that we typically always start with the problem and the benefits and the risks. So we start with the scientific data we have in hand, and then we add in all these additional layers and domains that actually reflect the complexity and influence our decision-making. In this instance, we have actually started here with the values, acceptability, resource use, and feasibility. And we have been waiting and waiting for that phase 3 trial data to come in. So we haven't been able to deliberate on the benefits and risks yet, which is why we can't make any clear recommendations until we have that phase 3 trial data in. And with that, I'll end. Thank you.
Thank you very much, Grace. We will try and do like a rapid fire answers to some of the questions. Thank you so much, Dr. Dekker, for actually already answering some of the questions that came in through the Q&A. I think one of the questions that we would we should just address up front is any information about when this might roll out to children, the vaccines, and where are we in that process, if at all we know. And I can open it to any of you to take that as a stab at that.
So I can just say that at least I know for sure that one manufacturer, and I think others have also stated at ACIP that they are planning to do clinical trials in children. I think the timing is a little bit more up in the air on that, but at least I know some of them have announced that they will be doing trials in children.
The majority have said so. I think the main thing is that we really need them to enter phase 1-2 trials. And I don't believe, unless Corey knows of anything or Nikki, I've heard of any that have entered phase 1-2 trials. And we need that because you can't just launch into phase 3 trial, I think.
Well, I think there'll probably be some bridging studies with the older kids and then eventually move down to the younger kids is my estimate.
The trials do go down to the age of 16, I believe now.
Yeah, Jensen's.
That's the youngest. But we haven't hit anything close to the school age population yet.
Right.
Thank you. There's a question for you, Grace. Will ACIP provide recommendations regarding monoclonal antibodies like Regeneron, since these are being evaluated for prevention as well, similar to Synagis?
It's a really great question. That actually, I have to say, it hasn't come up, and in part because I think we've been so focused on all the vaccines that are coming through. We do sometimes consider biologics, but that's not been one of the things on the docket for us.
Perhaps Corey, any comment on AstraZeneca's study on hold in the US and not in the rest of the world? Is there anything we can infer from that?
Honestly, I don't think so. You know, there are different regulators involved. The rest of the world has their own regulatory bodies that meet with the developer. I don't know what the holdup is in terms of the US in particular. I just don't have any information, and those are things that just happen between AstraZeneca and FDA.
Thank you, Corey. Tagging along the immunizations in children question, is it safe to assume that adult immunization will be rolled out before we know anything about kids?
Yes. Yeah. I mean, we can't consider a recommendation for kids without any evidence, even in the adult population yet. So agree with Dr. Klein that we will learn a lot from the safety and effectiveness in the adult population, but I think there are some unique considerations in kids, in particular with safety, that we're going to have to consider. So I imagine that we are going to have to rely on phase 3 trial data for that.
Yeah, I think there's another issue that is, if we are really, really lucky to get a biologic marker of efficacy out of the clinical trials, and that would certainly make expansion into some of the other populations much easier, because you would just target getting an equivalent response in kids as was shown in protected adults, for example. It may take us longer to get there if we don't have the full data from the trials, though.
One of the interesting things, I'll just chime in, is that the size of these trials are huge.
Yeah.
So they're, each of these trials, 30 to 50,000 per trial, gives us a huge amount of data on efficacy and safety. I mean, typically vaccine trials are much smaller in size and they take much longer. The size has been, out of necessity, because in order to be able to determine efficacy, they have to have enough people who might be exposed to infection. But I also think it's a benefit for safety. As long as we're able to, follow up on the data from those trials, we will actually probably have more pre-licensure data on vaccine safety than we ever typically have. I haven't seen trials this size since rotavirus vaccines, but, I don't know, Corey or Nikki, if you've seen other similarly sized trials.
I mean, I think that's a good point about them being much larger. The difference being that we're the expectation that there's going to be rolled out much faster, so we just don't have the time, because we're not going to be waiting for the full 2 years, for example, for all the data to come through before I mean, in assuming that there's some sort of efficacy, that before that they're actually rolled out to the public at large. And so, you know, in a typical cycle, as Corey explained, you know, nicely explained, all of that data would be complete before the licensure application goes to the FDA, and we would have that data in hand. But now I think we'll be generating it at the same time that people are being vaccinated, which I think in this case makes the post-licensure vaccine safety, you know, real-time monitoring that much more crucial in terms of trying to monitor and find out what's actually happening.
Thank you. Just a quick one. I have heard that COVID vaccines may help with severe disease, but may not affect nasal carriage. Is that true?
Well, I can go and try. I don't think we have any evidence to say that right now. I think that we just don't know if that's true or not at this point, and I'll let others people chime in if they have anything else.
The data that we have are for primates, and so there are some primate data where there are differences in, but those are artificial models, and they don't always predict what human responses are going to be. So we'll have to get data from these trials.
And I think it's a really important question because, you know, right now, most of the trials are focused on symptomatic infection, you know, plus or minus a subset that are severe, which is really important. However, you know, our hope is that the vaccine candidates that are coming through will interrupt transmission, and being able to interrupt asymptomatic infection is going to be just as important. Right now, it seems to me most of the trials are capturing it through antibody response to capture asymptomatic infection. But going forward, I think it'll continue to be an important part. We're assuming it'll interrupt transmission, but that's based on symptomatic data.
Thank you. There's another question. How strongly are trials controlling for enrollees' use of other medications like antiviral steroids during the trial?
Well, I can answer. There are certain criteria. The trials in general always have very strict enrollment and eligibility criteria, and the trials, these trials, as well as is really quite typical for most vaccine trials, there's usually some exclusion for some steroid use, depends on this trial, it depends on the amount of steroids, but something around the order of more than 2 weeks of steroids typically would be exclusionary. Antivirals also depends on the trial. I don't know if there's anything in these particular ones speaking to antivirals. I'm assuming you mean, well, I mean, remdesivir, for example, you can't be using, you can't have a history of using remdesivir, for example.
Mm-hmm. And there was a question from one of the participants who had to leave the conference, and so wanted to make sure we answered the question. And because these answers will not be available with the recording, I'm just going to point out, she had asked about storage and temperature considerations, and Corey had answered the question to say that DNA and RNA vaccines will be a problem because they require extremely low temperatures for storage, but other candidates should be okay at refrigerated temperatures. So these might be particularly a problem for low resource countries. Mm-hmm. Well, clearly it's a very important topic with a lot of questions, and we couldn't get to every one of them, but I think we got to most of them. I just want to thank our speakers for your time, for your expertise, and for sharing this very important information with us. And thank you to all of our participants for joining us. And as Alan pointed out, next week it's going to be about school. So we would love to have you join us next week. Thank you very much.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.